So this is the work that I've done throughout my time in the Shoulders Lab at MIT in the Department of Chemistry on commercializing directed evolution in mammalian cells. So taking a pretty broad look, every biomolecule in every cell of every organism that's ever lived has been generated through the process of evolution, which is pretty remarkable because natural evolution is undirected. There's one goal in an evolutionary system, and it's to survive, to pass your genes on to the next round, to the next point in time. But humans cleverly can direct evolution. You see this in crop domestication or livestock domestication, and the advent of genes and genetic biology has created a new type of directed evolution where humans can evolve a specific gene for a specific purpose. And examples of these evolved proteins include new fluorescent proteins, new therapeutic antibodies, and gene editors. And this is a really powerful technology that allows for numerous applications, but most of these experiments are performed in bacteria and yeast and not performed in human cells. And unfortunately, what occurs is that the proteins involved in these bacteria and yeast still function in the mammalian cellular environment, ultimately because you're evolving and optimizing for activity in the incorrect space that you want it to ultimately function. This is problematic for things like biomedicines or biotechnologies that you ultimately want to use in the mammalian cell environment. And so the obvious answer would be to do the evolution in the mammalian cell itself. Unfortunately, that's really tough. And I'll highlight three main reasons why direct evolution in mammalian cells is so difficult. The first is that the mammalian cell genome is huge, much larger than bacteria or yeast, and that means there's many escape pathways and that they're relatively difficult to engineer. The second is delivery. So delivering your genetic variants to mammalian cells can be fairly low efficiency, and then you can deliver potentially problematically multiple variants in a given cell. And lastly, mammalian cells are really slow to grow as compared to bacteria or yeast. Mammalian cells can take 1 to 2 weeks to expand in a population. All these things can really ruin a direct evolution campaign. And so our innovation was, okay, let's use a really fast evolving system to do this, to enable us to do a direct evolution circuit in mammalian cells, and that's viral evolution. And so what we do is we take our initial gene of interest and we encode that gene of interest in a human virus, and in infecting human cells, we simultaneously create a massive library of variants and select for new function and enrich the best performers all at the same time, and ultimately we identify the winners, these evolved biomolecules. And so our platform, we've been awarded a patent, an allowance, a notice of allowance on the methodology and the compositions of matter. That's the virus and the engineered cells. We've published our platform. Check it out in chat. And we're pursuing commercially valuable targets, in particular gene editors. And so what our ask for you, what we're interested in looking for from you all is expertise in where you think that engineered proteins in mammalian cells could be best used. We've had a lot of great conversations already today and last night to sort of build out sort of where we want to go next. It's a really exciting new space of technology of protein engineering, and I'm really excited to see where it comes forward. And come chat with us, find us by the poster, and thank you so much for your time.